Okay, so welcome Wednesday, March 1st. Next week, we're going to plan on being back in class at Elkhorn at 6:05. So plan on that. Um, I'll also attempt to open Zoom from the classroom. Sometimes it works. Sometimes it doesn't work, the recording if I'm moving around too much. So, but anyway, if you can't make it next Wednesday, I will attempt to pull up Zoom. In fact, it'll open, but I can't guarantee the quality. But I'll have my microphone. I'll bring my microphone, and it picks up pretty well. So anyway, I'll send a reminder next week, probably on Tuesday or Wednesday morning. So anyhow, we are, according to the map, starting. Let's see. Let me pull that up. Up. So syllabus, and again, you don't. This is a work at your own pace, so you can, if you're not where the calendar says we are, that's not a big deal. You have until April 21st, which is almost 8 weeks. So you still have quite a bit of time. I graded today, and most of the grades are up through at least unit 4, 3 or 4, somewhere around there, which is perfect. So this week, 3-1, we would be beginning unit 6, the immunity unit. So tonight, though, I do want to go through a little bit with case study 4 first, a couple of muscle identification, kind of anatomy, a little microanatomy update for you, or review. That's how this first one begins. So let me zoom in. Yeah. So you're an intern in rare musculoskeletal and neuromuscular disorders, and a group of high school students is coming in for a tour, and you've been asked to prep some info. So the first thing that you're tasked with is matching all of these muscle terms with the definitions. So you can go through and do that if you haven't already. Again, you can highlight, well, sodium will give you a whole bunch of options, but let's do like sarcoplasmic reticulum. You can highlight it, right click, and then hit search Google for sarcoplasmic reticulum. And it'll give you a definition, and then you can match that up. Or you may remember some of these items and be able to do them from that way, from memory. So anyway, take your time on that if you haven't already. And then we actually get into a neurotoxin exposure. So we've got a biochemist in the CDC developing a neurotoxin using sarin as a model substance. So you'll go through that. We've got Jeff. He's been brought in, dizzy, heart pounding, shortness of breath, incredibly sluggish. So Jeff is not doing well. So the first thing you'll want to do certainly is go through and look at and document some of Jeff's symptoms, which again are laid out right there. So just a second, I need to. Okay. So anyhow, so Jeff, he's dizzy, chest pain, shortness of breath, cramping, spasms, fatigue. Yeah. So colleague from across the hall finds him, calls for help, give him oxygen, get him to the hospital. All right. So then I'll ask, what's the role of acetylcholinesterase, or ACHE? Remember, it's ACE at the end, so we know that that's got to be some sort of enzyme. So it's going to break down acetylcholine potentially in the NMJ, which is short for neuromuscular junction. So, so right up here, you probably had acetylcholinesterase. So you've already uh, matched ACHE with the proper definition. So you can you can go right off of that, or like I say, come over here to Google cholinergic enzyme, so acetylcholine, and cholinergic. Generally, we would think is in play during a parasympathetic uh, bring down. So bringing you back down to normal ranges. So that's acetylcholine, or a cholinergic response. It's going to relate to parasympathetic response. Okay, so keep that in mind. Remember, you're either going to be sympathetic and adrenergic. So heart rate goes up, respiration goes up, all of the muscles start activating. You get that fight or flight action going. That's adrenergic. So when we get acetylcholine involved, we're thinking more of what we call a cholinergic. And then within the term acetylcholine, you can see choline. So anyway, that's going to be your clue if you ever see this on some sort of exam. So anyway, acetylcholinesterase found at the postsynaptic neuromuscular junction. So at the NMJ is what it's asking you. What's the role of acetylcholinesterase at the neuromuscular junction? So it's an enzyme. It has ASE at the end. And typically the ASE is relating to whatever comes in front of it. So if it's acetylcholinesterase, the ACE, or the enzyme, is breaking down acetylcholine. So anyway, it's an enzyme that breaks down ACH, or acetylcholine, at the synaptic cleft. And during a stimulus from the nervous system, so N for neuro, so the stimulus is going to come down the neuron, and we're going to get to that presynapse, or that little kind of knob or bell that of the nerve that connects to the muscle. They call it the neuromuscular junction. That bell is going to contain, or that presynapse, or that knob, or that axon terminal. It's got 10 different, at least, names for it. So axon terminal is used a lot. Presynapse, or presynaptic knob. So within that, you're going to find little vesicles containing acetylcholine. So when a synapse or a trigger gets sent down, or a stimulus down the nerve, it's going to send acetylcholine out of that presynapse, or that knob, that axon terminal. It's going to dump acetylcholine into the cleft, or the synaptic cleft, and then it's going to travel to what we call the postsynaptic region, which is going to be the muscle. So that's to get the muscle to contract. That's one of the first steps, because in order for a muscle to work, you have to have nerve plugged in and chemicals released. So acetylcholine is the chemical that's released to attach to the muscle fibers and get more action going regarding calcium release from the sarcoplasmic reticulum. So anyhow, when we need a muscle to relax, we need acetylcholine to get taken back to the presynapse. So acetylcholinesterase is the enzyme that's going to take that acetylcholine and break it down and stop the nerve signal from sending more acetylcholine to the muscle cells, or the muscle fibers. So anyway, we see acetylcholinesterase active when we were getting a muscle to relax. Once the stimulus to the brain, from the brain down the nerve out to the muscle, is saying we're good, we don't need, the bear's not chasing us, or we need to relax the muscle now, that's when ACHE kicks in. All right, so anyway, kind of a long winded answer for that, when really just put the definition of ACHE there and you'll be good. All right, so we admit Jeff, we've got some normal ranges, so we're going to look and see where he is. Oh boy, right away we see his blood pressure is awfully low, 105/65. Hematocrit is right where it should be. And now remember, hematocrit has to do with packed red blood cell count. So, and red blood cells transport oxygen. So if we have a really low hematocrit, we're probably dealing maybe with an anemia situation or something along those lines, where we don't have enough red blood cells to deliver oxygen, or we have an excess amount of plasma. So we just see the percentage get shifted. So I may have enough blood cells, but the percentage of red blood cells relative to the total blood volume may be lower because of excess plasma. So, but anyway, it doesn't matter with this situation because he's right where he's supposed to be. Glucose in the blood looks good. And again, that's important because along with oxygen transport, we need glucose transport to the cells so we can get some ATP. Sodium levels look great. Potassium looks good. Ooh, now here's where we're struggling. Well, let's skip that one real quick. We'll come back because that's the elephant in the room. So anyway, thyroxine levels, T4. Remember, from the thyroid gland. So a metabolic chemical or hormone, and then we have T3 levels, which again also these from our last case study, or case study 2, I believe it was. We talked a little bit about the thyroid and these two, and those levels are fine. These are very high when we're dealing with some sort of extreme situation with sympathetic or stress hormone release. So anyway, let's go back up here to the ACHE, right? This is what we're targeting. Normal range was 100 and Jeff's sitting at 44. So he doesn't have enough acetylcholinesterase to bring back or to stop acetylcholine from triggering muscle contractions. So he's gonna be having some, well, I don't want to give it away. We'll keep going. Which of Jeff's levels are abnormal? So primarily, right, his blood pressure's low. But it's not horrible, right? 105, look at the normal range, 90 to 120 for that systolic. So he's normal, he's low normal. All right. 65 for diastolic. Again, normal range 60 to 85. So he's in that range for diastolic, but still relatively low. So we don't need to put that, because he does have, it's low, but it's still within the normal range. So the only one that jumps out really as being a problem is that ACHE percentage. Okay, so just put that ACHE percentage. So now the next one, you'd have to do a little Googling, unless you already know what sarin is as a toxin or a neurotoxin. So if you do look it up, you'll find that, well, first of all, we know Jeff's working with a sarin-like chemical at his office. Secondly, he's showing signs of acetylcholinesterase blockage or inactivity or inability to be released, or something wrong with acetylcholinesterase. So if you look up sarin, you'll find that it's going to irreversibly. So that one, if we don't get medical attention quickly with something like this, your body isn't going to fix it itself. We have to intervene. But what it's going to do is irreversibly bind and inactivate that acetylcholinesterase at the synaptic cleft. So excess levels of acetylcholine start to build up at the postsynapse, at the muscle cell or the muscle fiber. Okay, so serin is blocking acetylcholinesterase, which means acetylcholine is just going to continue to dump into the muscle and cause the muscle to want to contract. So let's keep her moving. How would exposure to sarin like poison affect the amounts of sodium going into the muscle cell? Well, again, when we've looked at or you've looked at, hopefully you've watched the video, but if not, you will at some point. But one thing we know about the kind of dominoes falling to get a muscle to contract is that first spritz of chemical from the neuron or the synaptic knob is going to, the presynaptic knob is going to be acetylcholine. It's going to go to the muscle, and the second chemical or the second domino to fall is going to be the sodium domino. So if I have an excess amount of acetylcholine, I'm going to have sodium triggered as well. So I would have an increase in acetylcholine leads to an increase in sodium release as well into the muscle. Okay. So if I have a sarin or sarin-like poison exposure that inhibits acetylcholinesterase from being active, acetylcholine is going to build up at the muscle cell. It's going to then trigger an excess amount of sodium release. So you kind of see where this is going. So if you know, remember with the basics of pathology, you have to have the basics of physiology and anatomy, and especially microanatomy, at least under your belt. Because if you know how it's supposed to work, the basics of acetylcholine first domino, second domino sodium, third domino calcium. Then we get finally into the actin and myosin binding. To unbind or to relax the muscle, we have ATP being released to break that actin myosin bond, and then we set the dominoes back up. So calcium gets set back up first, then sodium gets set back up, then finally acetylcholine, and now we have relaxation of the muscle. But in order to have that, we have to get acetylcholine, the first domino. We have to get the enzyme activated so we don't have too many acetylcholine dominoes piling up. So anyway, too many acetylcholines means too many sodiums, means too many calciums. And what that's going to do is trigger intense muscle contractions or spasmings or crampings. Okay, so what number are we doing here? Number 5. So how would exposure to sarin-like poison affect the amounts of sodium going into the muscle cell? Explain why. So again, a sarin or sarin-like poison is going to drastically increase sodium levels going into the cell. Sarin or anything with the same mechanism of action, remember, inhibits acetylcholinesterase at the neuromuscular junction or at the NMJ. If I have a decrease in acetylcholinesterase, there's prolonged exposure of acetylcholine at the muscle cell. And those receptor sites on the muscle cell or the muscle fiber for acetylcholine are going to be extremely full. They're going to trigger an excess amount of prolonged depolarization. So remember, when we're at relaxation, we're polarized. When we are getting some sort of muscle contraction and a chemical release, we're going to see depolarization. So we're going to see a flood of sodium ions going inside of the cell in this particular situation. So all of that sarin that eliminates acetylcholinesterase triggers acetylcholine to build up, which triggers then excess depolarization, triggering all of that sodium to go deeper into the cell, or what we call an excess of sodium influx. So, okay, again, if you so you only have to know three dominoes. Acetylcholine's the first one, sodium's the second one, calcium's the third one. If you know that order, this is smooth sailing. Okay. And again, there are a few other chemicals involved. We mentioned ATP, and magnesium is involved in this at the deeper cellular level of that muscle fiber. So here's your third domino. You can answer this the same way you just answered number 5. How would exposure to the sarin or sarin-like chemicals affect calcium levels inside the sarcoplasmic reticulum? So, well, or inside the sarcoplasmic reticulum, we're going to see a decrease in calcium inside the sarcoplasmic reticulum, right? Because sarcoplasmic reticulum is where calcium is stored when the muscle is not being stimulated. In this situation, we have hyperstimulation of that muscle because of all this excess acetylcholine building up, triggering an excess of sodium, which then leads to calcium rushing out of the sarcoplasmic reticulum. So calcium levels inside the SR are going to be low. Okay, so because the sodium kicked it out. When a muscle's at rest, sarcoplasmic reticulum is full of calcium. Okay? When the muscle's not at rest, when it's being stimulated, calcium it gets dumped out of that sarcoplasmic reticulum. Okay? So we'd see a dramatic decrease in calcium levels inside of the sarcoplasmic reticulum. And the thing is, once a muscle relaxes, once we put the calcium domino back up, that requires calcium pumps. So, if it's a pump, it requires more ATP. So, but they can't keep up because all that calcium's getting dumped out. And so any calcium that's trying to go back in against its concentration gradient, the pumps can't keep up. So we just have calcium. Once it leaves the sarcoplasmic reticulum, remember, it binds to the troponin complex, causing the tropomyosin twist, and then we get exposure of the myosin binding site. Okay. So anyhow, that's number 6. And then there you go, number 7. How does the increased calcium levels affect the position of actin and myosin filaments? So again, calcium gets released to expose the binding site for actin and myosin. So if I have an excess amount of calcium, I have excess exposure of myosin binding sites on the actin, which then causes the triggering of a muscle contraction, or the completion really of the muscle contraction. So with additional calcium, again, those myofilaments, actin and myosin, are going to be in a fully contracted state. Okay? That myosin binds to the actin because of the presence of calcium. Calcium binds to the troponin and tropomyosin and shifts everything, exposes the myosin binding site. All right, so, and again, when you do the beginning of this as kind of a review of your muscle microanatomy and microphysiology, this all lines up. All right, so what needs to happen to Jeff's postsynaptic membrane to remedy his paralysis? Physiologically, what do we need more of? So this is where we started. This should be a slam dunk, straightforward answer. He just needs acetylcholinesterase. He needs the enzyme to stop all of this acetylcholine. Remember, that's the first domino to fall to trigger all of this other behavior. So we need Jeff to get more acetylcholinesterase. So, all right. So anyway, we're just about there. So that's what Jeff. So Jeff's done. Okay. Now we've got Sandy. We've got a couple questions with her, and hasn't been feeling well. Tired, coworkers, droopy eyelids. So droopy eyelids. Remember proprioception of muscle, so not involuntary muscle control is called proprioception, and that's going to keep the eyelids up. It's going to keep the cheeks, even when we're not expressive. It still keeps our facial expressions from drooping. So if we do have a patient that has drooping eyelid or lids, could be one or the other, or they have droopy cheeks or saggy one side of the face or the other. When they're at rest, that's a signal that proprioception is not functional. So anyway, let's see if that's so. There's a key term in there, droopy. Additionally, she's experiencing weakness in her arms and legs. So we've got some weakness going on, some more muscle issue, difficulty talking clearly. Again, the ability to speak comes from a lot of muscle coordination within the mouth and the throat area. So if she's having difficulty speaking, more muscle control problems. Parents success hasn't been looking very happy. So maybe kind of frowny even. Again, if you lose proprioception, it can look like you're mad or frowning all the time. Now she's choking at work. Again, swallowing not functioning. So we obviously have some neuromuscular issues with Sandy, and they're looking like they're systemic, meaning they're affecting multiple regions of the body. They're not isolated to just her face or her head or her throat. She's having arm and leg issues as well. So we need to get Sandy in. All right. Could be something that's very simple. It could be something catastrophic. We have no idea. So she comes in, what do we do? The typical vital signs and run these same kind of tests. So blood pressure looks good, right? 115/73, that's perfectly fine. Everything else looks really nice, right? I'd say we're sitting pretty good here. So all of these numbers, all of these Sandy results match or fall into normal ranges until we get to the last one, antibodies for acetylcholine receptors. So acetylcholine receptors, again, are going to be found at the post synapse. The receptor means it's receiving. What is it receiving? It's receiving acetylcholine. So the post synapse at the surface of that muscle fiber, the muscle cell, is where ACH receptors are going to be located. So every time I need a muscle to contract, even through proprioception. So with our facial expressions, particularly our posture, those are major proprioception, involuntary muscle contractions that are really kind of ongoing. So my point is acetylcholine's constantly being spritzed at low levels to maintain proprioception. When we need actual voluntary movements, we would see an increase in the release of acetylcholine at that point. So an antibody would be something going against potentially these ACH receptors. So this is not good because we generally should not find anything going against acetylcholine receptors. Those have to be open, available, and ready to receive acetylcholine all of the time. So we would not see antibodies that are going against acetylcholine receptors unless we have a problem. And that's what we see here. So let's find out what's going on. Symptoms, we just went through all of those. You can certainly, you know, go back up and fill those in. Or just like write them in: fatigue, droopy eyelids, trouble speaking, their difficulty speaking, difficulty swallowing, arm and leg muscle fatigue, lack of facial expression, So yeah, a lot of muscular stuff that's just not doing what it needs to do. So we look at her blood and we see again that antibodies are present for acetylcholine, and that shouldn't be. So number three, how would these antibodies against acetylcholine affect the neuromuscular junction? Well, they're going to inhibit the ability of these receptor sites to actually receive acetylcholine. Remember, acetylcholine receptor sites are going to be on the surface of that muscle membrane, right? The muscle fiber or muscle cell. Okay, we call that the postsynapse. That's where, again, acetylcholine stored in the presynapse of the nerve gets released and then travels to the postsynapse of the muscle. So it's the neuromuscular junction or the synapse. A synapse is where a nerve and a nerve join, or a nerve and a muscle join. In this case, nerve and a muscle. So the postsynapse is where these receptor sites are. They're ready to receive. Once they receive that acetylcholine, that's going to trigger the second domino, sodium from before. So, and then the third domino, calcium, which then is released from the sarcoplasmic reticulum. Then we get the binding of the actin myosin, we get a contraction. We're not getting muscle contractions, muscle control, muscle response from our patient. So, from Sandy. So how would antibodies? So anyway, so number three, the antibodies against ACH receptors, again, they're going to inhibit acetylcholine from attaching to the postsynapse or to the muscle. Thus, is going to inhibit the triggering of a muscle contraction. Now, is this like all or nothing? It's a complete shutdown? Probably not. The antibodies aren't gonna. There are gazillions of acetylcholine receptors on just one muscle. So the antibodies are likely not completely covering up. So she's able to get some chewing. She's able to get some swallowing. She's able to get some speech, some facial expression, some muscle movement, right? So it's not an all receptor sites are done. It's not enough of them. The antibodies have attacked enough of them to cause her to have some noticeable issues with basic muscle control, movement, and proprioception. So, how would antibodies against the ACH receptors affect sodium? So it's kind of going back to the same thing. So again, if I can't get acetylcholine in, I'm going to have a decrease in sodium. Okay. So then, how would antibodies? You could probably guess what the next question is without even scrolling down. It's going to be the calcium one. So calcium levels are going to be high inside of the sarcoplasmic reticulum because nothing is triggering calcium to leave. So this is the opposite of Jeff, right? You guys are catching on to this, I'm sure already. So Sandy must have some sort of specific disorder. So how would you find out what that disorder is? You probably type in antibodies. Here, let's do it. So it asks, what disorder does she have? Well, we know she has antibodies against ACH. Well, the ACH receptors, technically. And there you go. Pops right up. Disorder, antibodies against ACH receptors. Myasthenia gravis, the most common disorder. So we could then associate with that. So we could certainly think that that might be the issue. So what disorder does she have? How do we treat or manage the myasthenia gravis? So antibodies are involved. Where do antibodies come from? Where are antibodies produced? The immune system, certainly. So She has a hyperimmune system somewhere. There's a hyperactive release of antibodies against ACH receptors. So she has an autoimmune disorder. For some reason, her self cell, her body is recognizing acetylcholine receptors as being foreign cells, and so or non-self cells. Okay, so because uh, the immune system's recognizing ACH receptors as non-self cells, the immune system response is we need to fight against this. Okay? So antibodies get produced, they attack these receptor sites. So the thymus gland is one of the primary T cell T cell lymphocytes are coming from there. Natural killer cells are coming from there. We have a lot of action from the thymus. So, is it possible that the thymus gland needs to be suppressed? Maybe needs to be removed? That's pretty extreme, but as we age, we kind of lose our thymus gland anyway, or we don't lose it; it shrivels up. But anyway, maybe some glucocorticoids would help, some sort of steroid treatment. Um, Glucocorticoids will cause immunosuppression. So it's possible that with the glucocorticoids introduced, we might get some immunosuppression because that's what we need. She's got hyperimmunity. So, regardless, Sandy needs more testing likely. Initially, it'll be kind of a trial and error type of deal. And you do not have to do this table, by the way. So do not do the table. You don't have to worry about it. We just went through comparing all of that. So anyhow, that is what's going on with Jeff and Sandy. Again, some of you have already submitted that and it looked great. Um, okay, let's see. Me. Let's go right into this one. Now, this particular this case study is not very. We're not going to need to spend very much time on this one. Case study 5. This is going to be kind of an activity on the basics of DNA structure, as well as the history of the kind of understanding of the basics of DNA that occurred back in the 50s. So it's kind of a DNA history lesson, maybe toward the end. And the beginning or the first part is going to be more of a what are the basics of DNA. So it sets the stage, autumn 1952, discusses several details regarding the discovery, if you will, of DNA. So you want to read through that. How do cells make new copies of DNA? Before you can figure out how DNA is replicated, you must understand the structure of DNA. And again, you've probably all heard of and seen DNA at some point. So your levels are going to be varied regarding your previous experience with understanding DNA. So I put a video in here. It's short, but it's extremely jam-packed with great info. And if you watch this video, I'll pull it up. got a lot going on Zoom with Zoom and recording and Windows. So I'm not going to play. It won't play. It might play, but it might kick us out or something weird might happen. But there's the video. Um, there's a transcript of the video if you want to just read through that. That's certainly an option. So you can click that. It'll just print off a PDF of everything they're talking about. So from the video that you watch or from the transcript that you read, you'll be able to, oopsie, you'll be able to answer these basic multiple choice questions. Okay? There are only 8 of them. Um, so anyway, if you read the transcript and or watch that video, the link that I put there, all of these will be slam dunk. Okay. After that, you'll dig a little deeper. That won't take you too terribly long to do, but while you're doing it, kind of you'll be either refreshing your memory or gaining a stronger understanding, either way, of the basics. So from there, you're going to get into a little bit of the history. So there's the actual paper from Watson and Crick, who are credited with the discovery of DNA. Whether or not it was just those two or there were other players, that's what you're going to find. There's an annotated version as well. I also put a short, this is an 8.5 minute discovery of DNA video, very informative as well. So you'll want to check that out. And then the structured DNA website gives you a little more detail as well. So I want you to check all of these out. And then once you've done that, he'll be able to go through and answer these questions. Why is understand? And some of them are kind of subjective. I mean, you kind of your opinion based on what you know. Others are there are specific answers that you'll want to do for those. And then a little bit about what each of these individuals did regarding the discovery of DNA. So this is kind of an activity of understanding the basics of DNA, understanding why it's important to know the basics of DNA and how it's used today and in what areas, and how important this discovery was, but also to go through, we're looking at 70 years ago or so, 71 even, 70 plus years ago, really how the science world was operating. So versus today, and maybe there's some similarities, maybe there's some differences, but regardless, I want you to pay attention to some of the maybe cultural aspects of 1952 in regards to credit given to Rosalind Franklin specifically. So anyway, so you can touch on that a little bit if you'd like, but this is students generally like all of the case studies, maybe except for number 3 might be a little bit more challenging or whatnot. So anyway, this is another one that you'll enjoy doing. A little history lesson. So let's see. I wanted to. And again, if you have questions, just send me an email and we'll take care of it. Next week, I'm going to dive into case study 6, and I think we'll probably have time. We may get into case study 7 as well. But case study 6 is one that I want to go through. And like I say, I'm going to be on campus, and I'll also Zoom and attempt to record that as well. Next week's is immunity, and this one's on COVID-19. So it'll be a pretty interesting one to discuss and to go through. It'll provide you with a little stronger knowledge of the basics of immunity, particularly viral immunity. So anyway, that'll be a good one to discuss and very relevant. So that's probably all we'll have time for next week. Now the other one I think most of you have submitted, but I do want to go through a couple of things. Again, I put a bunch of links in here that may be helpful as well. Let's see. I really just wanted to pull up the questions there and just go through and kind of answer these kind of along the way. Test results, when we looked at those, there are a couple of possibilities, I would say. The one we really are kind of looking at in her situation is going to be hyponatremia. Now, you see that word hyponatremia, and you see the hypo, meaning not enough of, right? Below normal. And again, blood pressure and temperature a little high. Some of you maybe put that, that's fine. They're not so far out of range, though, that we'd have Brittany behaving the way she is. So her bigger issue is going to be sodium levels, or water levels. So hyponatremia. So we've got emia in that term, so we know it's a blood condition, and hypo, we don't have enough of something in the blood. So Na, remember, is the chemical symbol for sodium. So sodium is natrium in, I don't know, German, I'm sure is where that got that name, natrium. So, but we call it sodium here, but we kept the Na portion of natrium. So anyway, hyponatremia, not enough sodium in the blood, which generally sodium is in fairly high concentrations in the bloodstream or extracellularly relative to this volume or percentage or concentration of potassium. So under normal conditions, we would see a lot of sodium outside the cell, a lot of potassium inside the cell. If I dilute the outside of the cell with an excess amount of water, it would cause the concentration of sodium to decrease. So that's what we have going on here. So if we have too little water, so not enough water, so in a dehydration situation, we might see something called hypernatremia. We'd have excess sodium in the blood. Okay, so let's see. Assume movement of molecules is limited, it can move to the opposite side of a container or stay where it is. If movement is random, so kind of like flipping a coin, it can go here or there. It can be heads or tails. Well, so you've got what are the chances that it's going to move to the opposite side? So I think most of you get this one correct. Just like flipping a coin, right? You've got a 50-50 shot. So we've got 50% chance or 50% odds that we're going to see the movement of the molecule across the membrane. So if we have 10 molecules, 5 are going to stay where they are and 5 are going to go to the other side. And again, part of that is to maintain equilibrium. Remember, we're trying to maintain equilibrium. So we want concentration gradients to be the same or as close to equal as possible. Okay. I just answered number 3. We've got 10 on one side. We're trying to get to equilibrium. What would you expect to happen? Right. We'd expect to see B. And we start to see 5 go across. Again, even distribution, concentration gradients. Behavior of the molecules is independent of the other molecules, so they're not talking to each other and asking each other what to do and what they think. They're just going away from each other generally and getting to, again, that state of equilibrium. So let's move on. I think that one's pretty straightforward. Number 4 just describes how these molecules behave over time due to random movement. So if we just let them sit there and let time pass, are we going to continue to get movement? Are they just going to stop moving and settle to the bottom? Are we going to get more going to one side, more going to the other? Okay. Generally, we'd probably see no letter C. All the molecules are going to move so that red and blue will become equal on both sides. Again, we're trying to, or we aren't, the molecules are uh, going towards maintaining equilibrium and having that concentration gradient be the same on both sides of the membrane. So over time, all the reds will go to one side, five reds on one side, five blues on the other. All right. Which of the following molecules could move through a phospholipid membrane with the least difficulty? So, least difficulty. Hmm. So let's take a peek at these items. Remember, everything's a solid, a liquid, or a gas. So, well, we have plasma too, but I'm sure there are other things. Anyway, but as far as we're concerned right now, we're dealing with solids, liquids, and gases. So I want to move a liquid across a membrane from outside the cell to the inside of the cell. So water, is that least difficult? Probably. It's not very difficult, right? But let's keep going. Glucose. How about moving glucose across phospholipid membranes? That's a solid, and that remember, glucose movement does require some sort of a helper, some sort of integral membrane protein that's going to be a channel or a receptor that's going to be able to grab glucose and bring it into the cell and so that's a lot of work, is I guess what I'm saying. So that's definitely not right. How about sodium? Sodium again isn't going to be that's a solid as well. That's also going to require some sort of integral membrane protein channel to allow sodium to pass through. So I guess so. It's a solid. So if I'm moving solids through the across the phospholipid membrane, it's going to take a little more than it is to move a liquid. Now, how about a gas? So they give us oxygen. Gas diffusion, moving from high concentration to low concentration, is the easiest, least resistant movement. So you definitely want to go oxygen with that. And then amino acid is again another solid. So that's going to be the most sluggish or take the most energy to move across the cell, or maybe not actual cellular energy, but which some do, right? Like glucose. Okay, so let's keep going. I think number six. We expect to happen over time in Brittany's cells. Inside, we've got 300 millimoles of salt or sodium. Outside, we've got 250 millimoles of salt. So, what do you expect over time? Remember, we're trying to get to equilibrium. So, let's take a look at our options. Water, let's see, where are we here? Water will move from inside to outside only. So so remember, we've got options when it comes to movement. We can either move the substance, so I could move salt out, or let's move salt into the cell to balance it out. Wait, inside, here we go. So inside there's more salt. So I could just move the extra salt out of the cell, right? And try to balance it out. The other thing, so that's going to be some sort of diffusion, where we get the substance moving, whether the solute is moving, right? All right. So remember the other option is we dilute. So how about I just dump a bunch of water inside of the cell and dilute it and then try to get some sort of equilibrium established regarding the concentration percentage or concentration gradient of basically the salt solution percentage to get those balanced. So yeah, I could do that. Water will move from inside to outside only. So is water going to move from inside to outside only? No, certainly we're not going to be diluting the outside. However, does water move from inside to outside a little bit? Yes, indeed, all the time. So we can eliminate that. Water will move from outside to inside only. Well, it makes sense. We would want to move it from outside to inside to dilute this excess amount of salt. But we have the word only again, and yeah, we don't only do anything generally. So both A and B are out. So um, again, B, if you'd answered one or the other, B would be your better answer, but neither one are appropriate because of the word only. Water will move in both directions, but more water will move inside. That sounds a little bit better. Water will move in both directions, but more water will move outside. No, we don't want to dilute where there is less solute. We want to dilute where there's more. Or water will not move. Yeah, that doesn't make sense. So we definitely are going to go with C because water is constantly moving into and out of a cell, and we're trying to establish equilibrium there. So, all right. Let's go on to 7. We start to get a little bit away from things here. 7. You add a draw. So these next few are kind of more or less hypothetical, kind of just basic questions that you'd maybe do in a lab. You had to drop a blue dye to a container of water. After several hours, the entire container is blue. So, right, we could do that. So you do that at home if you've got blue food dye or any color food dye. Then you have to be blue in your cupboard. You could take a glass of water, drop some dye in there, and after several hours, the entire container is going to turn blue. Which statement is correct regarding this process? All right. So let's see. We've got a couple that maybe kind of make sense and a couple that don't make sense at all, and one that really kind of sticks out. So high concentration of dye particles repel each other, causing the dye to spread out. So they're not really repelling each other. This almost implies that they are that there's like the ability for them to choose to be active and repel and go against each other. This is not what's happening. They're not. Don't dump them in and they just start fighting against each other and bouncing off of each other. That's what repelling means. So not really happening. Particles moved until they were evenly distributed and then they stopped moving. So so put the blue dye in, everybody kind of just disperses out. Remember, we go from where there are a lot of us to where there aren't as many of us. So that's what's going on here. And then ultimately we get that nice light blue color and then they stop moving. Not really. They are going to continue moving around. If they stop moving, then they'll settle to the bottom. Because remember, liquid is a fluid medium. So you've got this dye in there that's also a fluid medium, but a little bit different density. And so it's kind of just at it's just moving where the water, within the water. And as soon as another dye molecule gets close to that, then it's going to move somewhere else. And so they're constantly dispersing from one another. Let's see. There were too many particles crowded into one area, so they moved to an area with more room. I mean, I suppose to a degree it's kind of describing what diffusion is, but I don't know if there are too many particles crowded. That's not your best answer either. I don't think that it does make some sense, but it's not, that's not the whole story. So let's keep going. By random chance, dye particles move from areas of high, so this is more of a sciencey answer, higher concentration to areas of lower concentration. So this is kind of our first real answer that sounds sciencey. Let's go to the next one. Dye molecules were broken into smaller pieces. So we took a big chunk of dye and they just broke themselves into smaller bit. No, again, we've never discussed that. That doesn't even make sense. So I would say A and D are the only ones that really have this high concentration, low, type of thing. Higher, high. So that's usually where students select from, A or D, and some C's because that's kind of what's going on. But again, you have a better answer than that. So A says high concentration of diet powders repelled each other. So again, they're not repelling each other. That's like not physically getting into it here. Um, this one throws students occasionally, but this is the one. It's D, by random chance. So it is random. It's like flipping a coin, right? This goes back to one of the first questions in this activity. You flip a coin. Is it heads? Is it tails? I don't know. It's random chance. So by random chance, they move from high concentration for there's a lot of them to where there are less of them or fewer of them. Okay, so D makes the most sense there. Now, if we continue watching that container for several more hours, what do we expect to maybe happen? So let's see. Dye molecules stop moving. Nope. Dye molecules settle to the bottom of the container. And maybe over a long period of time, that may happen. Dye molecules will continue to move around randomly. Hey, wait a minute. That sounds a lot like what we answered right before, by random chance. So that sounds pretty good so far. Dye molecules will continue to move such that the container will become different shades of blue. So there's it's gonna be dark blue over here, light blue over there, medium blue down there. We know that's not happening. It's after several hours. Dye molecules will continue to move, but in a non-random manner. So they're going to be organized. They're going to line up, and you go here, you go here, you go. No, that's not happening. So let's go back to C. Dye molecules continue to move around randomly. They're going to move around randomly. As they're moving around randomly, they're still moving from high concentration to low concentration. Okay, what do we got here? Number 9. We want to know whether a specific molecule pass through a biological membrane. So will it pass through? What do we need to consider? Specific types of lipids present in the membrane. Do the lipids of the membrane have anything to do with movement of a molecule? Not really. Remember, it's intramembrane proteins, and these channels, they have a lot to do with moving things in. All right, B, the degree of which the molecule is water soluble. Yeah, that sounds pretty darn good to me. You know, remember, water soluble is the water is the majority of the fluid inside of the cell. It's also the majority in the fluid of the fluid outside of the cell. So the degree to which the molecule is water soluble is going to determine its movement. If it's not water soluble, it's not going to move into the cell. It has to be water soluble. Okay, so we when we want to know whether a specific molecule will pass through a membrane, it has to be water soluble because remember the majority past that membrane is water. So if it's not water soluble, it won't. So like a fat molecule. Okay? Whether the molecule is actively repelled by the lipid layer. Now, the lipid layer is a passive. It's not alive, actively doing anything. Whether the molecule is harmful to the cell. Again, this doesn't really have to do with whether the molecule passed through. We know very well that harmful molecules pass through cell membranes all the time, right? Viruses, bacteria, and hopefully they don't, but they do. And the inside of the cell then has to handle it or deal with it. So your best bet for number 9 is the degree to which the molecule is water soluble. That's holds true all of the time. Because if it's not water soluble, it just won't pass through the membrane ever. Versus D, the molecule is harmful to the cell. Well, many molecules are harmful to the cell, and they manage to make it through. So that really doesn't have to do with anything. All right, so last couple, when sodium ions cross a membrane. So sodium ions cross a membrane. They must move down the concentration gradient. So sodium must move down its concentration gradient. So must, all it's a sodium must move down its concentration gradient all the time. No, it does some of the time. But remember, we have sodium potassium pumps that pump sodium against its concentration gradient as well. So when we remember, we have a ton of sodium outside of the cell. We have a little bit inside the cell. Well, what happens when a little bit inside of the cell needs to go outside of the cell? That sodium is being pumped against its concentration gradient because it won't naturally diffuse from an area of low concentration to high concentration. So it's not going to go to where there's nobody around, where there's a ton of sodium around. It generally likes to stay where it is, where there are less sodiums. But sometimes we need to add it out or get it out of the cell, and it's not going to naturally do it. We have to pump it. So when sodium ions cross the membrane, they diffuse across the bipolar without any help. Well, that's true going in, but not coming back out. So we're talking both in and out movement of sodium. They are helped across by proteins. Yes, we like that. That's your sodium potassium pump going out and a sodium integral membrane channel going in. Sodium ions can never cross membranes. Whoa, no, hey, easy. Yeah, that's bad if that's happening. Okay, last up is 11. Let's see. Oops, I pushed the wrong button. Hopefully it didn't alter anything. There we go. All right, number 11. Swimming in the ocean, you cut yourself on a seashell. Seawater is a solute concentration of 0.3 molarity. The fluid in your body has a solute concentration of 0.27 molar. When one of your red blood cells enters the water, what do you expect to happen to the red blood cells? So you get cut, the blood leaves you and enters the water. So so we've got 0.27 molarity of the cell of the body, and then we've got 0.3. So this has to do with concentration rate. So this just means there's more salt. You're in the ocean, so there's more sodium inside of the ocean than inside of your body. So now what we've done is left the body. Now we're in the ocean. So now I have a blood cell that's now in the ocean. And it's not used to being in such a salty environment. So are we going to dilute outside of the cell? Or are we going to send more water inside of the cell, the red blood cell? So let's find out. Nothing. RBC's membranes don't allow salt or water to pass. We know that's not true. Water and salt will enter the cell. Water will enter and leave the cell, but more will enter. Where's all the salt? Remember, it's outside, it's in the ocean. Water will enter and leave the cell, right? Remember, water's always entering and leaving, but more will leave. That sounds pretty good. We'll come back to that. Water and salt will leave the cell. Maybe, but we have it for sure. Water will enter and leave the cell. Yes, we know that's a fact. More will leave versus more will enter. Remember, more is going to leave the cell because water goes to where there's higher amount of sodium. So we have a higher concentration of sodium, so D is appropriate. More water is gonna leave to try and dilute the extracellular space. All right, so hopefully that answers all of that. So again, water, I'm gonna go D there. All right. Hopefully that helped out. I know I've got a video in our course that talks about both case study 3 and case study 4, but you've got a good grasp on those hopefully now after tonight. And then case study 5, that one the DNA. That's just research a little bit about what's going on, and you'll zip right through that, and make sure you watch those videos that go with the DNA one. Next week, we'll be back in class. We'll discuss case study 6. We'll get into immunity a little bit. We'll talk about COVID-19 and other viral activity and how it affects the body. In the meantime, keep working on your Padlets. A lot of you have finished up the Padlets and the labs, so you want to make sure you get into the McGraw-Hill Connect and get that taken care of as well. And of course, keep working on your exams and plugging away. And I'll see you guys next week.